Good morning, everyone. My name is Carrie, and this is Nima, and there's too much feedback. There's my volume button. I'll start again. Can you hear me? Is that okay? So I'm Carrie, and this is Nima, and together we'll be spending the better part of the day showing you how to use the IDB search interface. So I'll start with a brief overview. The morning we're going to start with an introduction, so the homepage search interface. We're going to be looking at the results page and all the details there. Then I'll be telling you a little bit about some features called finders. Randy alluded to them, and we'll be explaining how they work and where they are located. Then Nima will be taking you on a tour using the specialized searches, which are all field searches. Then we'll have a little break and then come back and talk to you about the email browser in detail. And then we'll start in the afternoon with some example queries. We're going to do as much of this live as we can. We'll have lunch and we'll come back, and whatever we don't finish up, we'll finish up then, and then we'll give some participant queries. So the first couple of slides are essentially setting the stage before I jump into showing you how to use the search interface. The first thing I want to get at is I want to convince you that even if you're an informatician who's only interested in predictions, that the search interface can be very useful to you. So think of the database, and you've heard this from both Bjorn and Randy, as essentially two buckets. It's a bucket of tools, but it's also a huge bucket of data, as Randy and Bjorn have explained to you. So you can see there are hundreds of thousands of epitopes in there: T-cell, B-cell, binding data, ligand dilution data. So it can be used for finding epitopes. So these have been validated. These are tested in some host and shown to be reactive. You can use it for the big picture, what is known. You can also use it for validation in your predictions, and we'll be explaining that in a little bit detail because we do have both positive and negative data. So it helps you define the known universe, what has been done and what has not been done. And this is very important because if you are doing predictions and then going to synthesize peptides and write some paper, you want to know what is known in that field, and the database is excellent at allowing you to get at that information very quickly, very efficiently. So it may be a part of your prediction informatic pipeline. And what I mean by that is kind of the schematic. So you have a protein of interest, you run a search in the database, and you can quickly find out what the known universe is, right? Then you use the prediction tools. You're going to hear about this tomorrow. You'll have some data set that you generate. You're going to refine this, maybe filter it, and then maybe go back to the database and use the database of known tested things in humans or in mice to validate your predictions, because again, we do have negative data. So the next thing I want to touch on is jargon. IDB has jargon associated with it, and some of it has coming up, so how we define things, so it's very clear that it's good that we spend a lot of time with a little bit of background. So these can be immunological terms, these can be ontological terms, as Randy's been telling you about, and these are, the other jargon is kind of database necessity, because we have to be very explicit, but we have to be very concise. So examples of this is we say immunogen, immunologically we say immunogen, that's what immunized the host, versus the antigen. The antigen was what was on the plate to demonstrate the immune response in the assay. Similar, we say assay. We don't say experiment, we say assay. So again, you're going to hear that all day long. We say in vivo administration, that means a mouse or a human was given something like a vaccine or experimental infection. Or in vitro administration, that means, for example, peptide restimulation in vitro. We say occurrence of disease, so that's a natural occurrence of disease, like they did have flu virus, they did have flu, or they did have type 1 diabetes. Similarly, we have in vivo processes, 1 and 2 to say that something happened in time. One happened first, and the second thing happened subsequent to that first one. Ontologically, Randy talked about this. This is database interoperability. This is so our database can link in and out with other databases, and these are relational things like parent and child nodes, and then siblings within those nodes. And then finally, database necessity. We say things like source antigen. What do we mean by that? We mean the antigen from which the epitope came. So it's very short, explicit, because everything's very epitope-centric in the database. Similarly, source organism, the organism from which the epitope came. And then we say non-peptidic or peptidic to mean non-protein. So you get kind of the idea. And all of this is available through the field description. We have a link here to the field descriptions and the curation. All right, so the next thing I'm going to talk about is, again, conceptual stuff, so to help you hopefully understand the search interface a little better. As you've heard in both Randy's and Bjorn's talk, There is some epitope, and that comes from some protein, and then that comes from some organism. So we are very much thinking in those terms, and the database is very much built around that concept. Similarly, in the assay, you always have a host, you have an immunogen, and you have an antigen. So the immunogen is something that immunizes the host, and the antigen is what was tested to demonstrate that response. The immunogen can be the epitope, it can be the protein, or it can be the organism. Similarly, the antigen can be the epitope, protein, or an organism. Excuse me, had too much coffee. All right, so without further ado, that gives you kind of an idea of the conceptual aspects of the database and how it's organized. So we're going to be talking today about this area, and you've seen this on both Bjorn and Randy's slides. So we're going to be talking about the main page search interface, as well as the specialized searches, and Nina's going to walk you through that. And then tomorrow, you're going to hear about the analysis resource. So I'm going to zoom in here to these search panes here. We're going to call them search panes. Each one of these is specifically designed to represent specific elements of the immune response. So an epitope is only an epitope in the context in which it was defined, the host, and whether it's a B or T, for example. So you get the context and the characteristics in this. The vast majority of the queries you're going to be running can be handled on the main search page. So I'll walk you through this. So you can look at epitope elements, whether it's discontinuous or non-peptidic. You can look at the type of assay, so broadly speaking, the response type, B cell, T cell. You can look at the antigen from which the epitope came, say it's flu, and maybe a hemagglutinin protein, the restriction that's defined in the assay, the type of host, and the type of disease. So if you click the search button, you'll find yourself landing on the results page, and you've seen this in several screenshots before. Now there's a lot of information here that's very packed with details, but let me walk you through it slowly. So the main part of this page is a summary table. So you have a summary table for each element of the response for the particular query that you're in. So you have epitope information, antigen information, information about the assays, any receptors if they were provided, and all the reference information. In parentheses is the total number of those for the particular query you ran. All right, so it's tab delineated, so you can click through the tabs. Each column is a field, and each row is an instance of data, all right? So if we look down now here along the left-hand side, you'll see a recapitulation of the search interface that was on the main page. And this is there to allow you to refine your initial query. If you get results back and say there's a great number of results, you might want to further refine that. Also, you can get at finders that aren't available on the main page, and also use this area to do the specialized searches, and Nima will be showing you that. So I'm going to zoom in here and show you two additional aspects here and here. Randy showed you this, but I'm going to emphasize it again here. So along the top, you have pending filters and current filters. This keeps track of your query input. So if you've forgotten which parameters you typed in, you can glance up there and that'll show you, and you can click this on and off, and I'll be showing you that. And then also up in the top right-hand corner, and it's also down at the bottom of the page, you can export all the data from each tab, so the reference tab or the epitope tab, in Excel, which is very handy, and I'll be demonstrating that later. So in the next couple of slides, I'm going to walk you through all of the search interfaces and show you the details and what they're for, all right? So we'll start with the epitope search interface. The default is any. That allows you to look for anything initial in your initial search. You can specify linear or discontinuous or non-peptidic using the radio buttons. You can also use the text field to type in or copy and paste in a peptide that you might be interested in searching for. You can do an exact match, or you can do a homology search. All right, so the next search pane you'll encounter will be the antigen search, and the antigen is the protein and organism from which the epitope came. So these can be pathogens like virus or bacteria. These can be self-antigens from mammals, or these can be allergens from grass or food, that sort of thing. And again, the text fields are set up so that you can type in, and here I did an example. I did HCV, and this shows you the autocomplete that we have. This feature is very much a Google-like feature that allows you to make your queries more efficient. All right, and yes?
So if you have more than one, you just comment, delimit them in the previous search. So if you want to look for two, like,
If you want to look for two?
yeah, you just.
Yes, and I can show you how to do that.
Okay.
Yeah, if you autocomplete, for example, if you wanted to do this one, you'll select it, and then you start typing again, and so you can multi-stack. And then there'll be a little number that'll show up, and that'll tell you how many you have.
Okay.
So if you have 3, it'll be a little 3.
Okay.
So yeah, and you can click them off and redo the search.
Oh,
It'll show
great.
you. Yeah, good
Thank you.
question. Okay, so the receptor search interface is a brand new feature that we're very excited about. We've been working really hard to get these data. Now that more and more papers are providing it. So it allows you to look at TCR or BCR sequence data. So we have alpha and beta, CDR 1, 2, and 3. So you can see here, if you check this box, it'll search for all the instances of papers that have BCR or TCR data. So you can look at the high level, at the type, so TCR, BCR. You can look at the chain type, heavy, light, alpha, beta. You can also look at CDR 3 on 1 and 2. You can also copy and paste in a CDR sequence and then do an exact match or a homology search, similar to how you would look for a peptide. So then the assay search pane allows you to look at the high level for all T cell, all B cell, or all ligand assays, so that would be elution and binding assays. You can unclick using the checkbox. The default is all, and the default is positive, but of course you can unclick and do any combination thereof. You can also use these search fields to type in, for example, IL-2 or ELISA, and you'll get, again, an autocomplete. You can also access finders, and I'll be demonstrating those very shortly. The next search pane is MHC restriction. This is restriction defined in the assay itself. So again, the default is all, but you can specify the class, so you can look at class 1 and class 2 separately. You can also type in a allele of interest, and you'll get an autocomplete, and you can select from that list. Next is the host. This is pretty straightforward. Most of the time, you're going to be using this to look for human data or mouse data. You can also use this text field to type in, for example, maybe an uncommon mouse strain or a very specific mouse strain, transgenic mouse strain, for example, and that will, again, get an autocomplete in that. So you can look at human data and animal models of human disease. All right, and then search by disease. This search interface allows you to look for the clinical status of the host. So this is not a categorization, so it's not, these are flu papers, these are diabetes papers. This is if in the patient history they documented that the patients had type 1 diabetes or dengue hemorrhagic fever. That's what this is for. And also known animal models of disease like EAD, for example, for MS. So we have the high level infectious disease, allergy, autoimmune, and transplant. And it's really nice because it allows you to look at differential reactivity if you want to see what has been done in humans versus what's been done in the animal model of that disease. So it's nice for that. And then lastly, at the very bottom end, as you're scrolling down the results page, is the ability to search by reference. This is just a snapshot I did from PubMed, which shows you can use all of these parameters here to search for paper of interest, the author name, the PubMed ID, etc. So that's pretty straightforward there. So I've now explained the search interfaces and how they work and what they get at. Now what I want to do is spend a little time talking about a feature that's behind these search interfaces, and this ties in nicely with what Randy has talked to you about. And this is the finders. A finder sounds pretty straightforward. A finder is a unique feature of the IDB that allows us to provide standardization and hierarchical organization by having collapsible data trees. And the nice thing about having these trees is you can look at the high level, but you can also look at very specific things. So we call that granularity. So you can look at the big picture, or you can look at a very granular aspect of the data by having these collapsible trees. And these data trees are built on either ontological or taxonomic organizers. So you have a parent-child relationship with siblings, as you see here. So essentially they're working behind the scenes, but they're also accessible to you. So they're trees that you can look at and peruse to see what data is there, because if we have encountered it in the data, it'll be in the tree, all right? So things, as Randy told you in her slides, aspects of the data that are compiled using these data trees are our protein trees, so our antigens, our non-peptidics, the organisms, the alleles, the assays, and the diseases. So you get a feel for that. So where do you find these? I'm going to take you through this. So you've seen them in many of the screenshots already. You see this little blue button here, so this is on the main home page. On the results page, once you've done a search and you are on the results page here, you'll see as you mouse over these search panes that are on the left-hand side, they pop open to reveal the Finder. So you click that. So in the next several slides, I'm going to be taking you through each Finder that you encounter as you go down the left-hand margin using these search interfaces. The first one I'm going to start with is a non-peptidic. So the non-peptidic Finder, so I'll walk you through. There'll be a little bit of repetition as I'm showing you each one, but as I go, hopefully I'll be able to specify just some things that are unique to that particular one, but I'll take special care in the first couple to describe the interaction here. So the non-peptidic epitope Finder, as Randy talked to you about, allows you to get at the non-protein chemical entities that the immune system encounters. These include metal allergens, drugs, lipids, glycolipids, those sorts of things, all right? So you would click the Find button, and what you'll get is this window appearing down below. And all of them are set up the same way. You have the ability in the left-hand side to type in an input, and then there'll be a data tree here, and then down below will be where the results of your search here will show up, all right? So you'll be able to click search, and you'll get that. And as an example, here I typed in cardiolipin, I hit search, and these are all the matches I got to cardiolipin. All right, so that's non-peptidic. Now we'll move down to the organism Finder. And again, this links to NCBI taxonomy, excuse me. When you click this open, you get this window again. Now, the organism, I want to emphasize again from the earlier slides I show you, the organism is the organism from which the epitope came. So if you're interested in influenza, or the immunogen in the assay, if it was the organism, or the antigen in the assay, if it was an organism, so you get an idea. So if you click Finder, you'll open this window, and in this case, I typed as an example Leishmania, and I hit search, and then I get this list of hits matching that input, all right? So then there's two things that you can do. So I'm now zooming in to the same window. So you can click either the plus sign or the highlighted tree. This yellow highlights where the input you put in lands in the data tree. So you can see the high node for Leishmania and all of the different species underneath, all right? And you can open those further to look at different strains. Alternatively, if you find one that you are interested in, you click that, it changes to a check. It shows up here in the current selections. You hit apply, and then it applies it to your search. So then you're back on the search page, all right? So this is just another example of a data tree. So for example, here's the dengue high node, and it has all the serotypes underneath, 1, 2, 3, and 4. And if you click open 3, it shows you all the children underneath that branch. All right. So you can look at the high node, or you can look very specifically. So it's a very powerful way to organize the data. All right, so the antigen Finder. So we were here. All right, so this links to NCBI GenPep, as Randy told you. So again, this could be the protein from which the epitope comes. This could be the immunogen in the assay or the antigen in the assay. So you click the Finder and you pop open. A little difference here, you'll note, besides the normal input and the tree next to it, is you'll see the ability to type in not just the protein name, but also the organism. So as an example, I did HA from flu, and I hit search. So you can see down below, I get hits matching that input. So in this case, I'm going to highlight it in the tree, and you saw this. This will be similar to what you saw in Randy's slide, and you'll see this high node of hemagglutinin with all of the different accessions gathered underneath it. As she mentioned, there are many, many, many accessions, individual accession IDs in the database. So it's organized under influenza A proteins, and it would be HA matrix and the other all 10 proteins there. And you can click that open and look at individual ones, or if you click the high node, you can get all HA from flu. So the receptor search interface, I want to indicate there's no Finder there. However, it's very simple. If you click the box, you'll get, and run a query, you'll get all of the information, all the data related to TCR and BCR receptors. Alternatively, you can click one extra criteria, input criteria there. I said, give me TCR alpha beta, and you get a subset of that data. So it's very, very streamlined. You can have very specific criteria or very general. And when you do that, as you saw with with Bjorn's talk, you get on the receptors tab on the results page, you get a summary of the data, so a group ID, which is an internal ID, the species that the receptor came from, what type, chain 1 and chain 2 of CDR3. And then this is also downloadable in Excel. All right, the assay finder allows you to look at a specific assay within the broader broader type of response, so a specific kind of T-cell assay or a specific kind of B-cell assay. In most instances, the autoclude page will find it, but if you're not able to do so, you can click the finder open and specify. One unique feature of this, again, it's set up in the same way, one really unique feature of this is the ability to look at the measurement of. So you can look by technique, so ELISA, ELISpot, but you can look for the measurement of. For example, you might be interested in looking for all gamma interferon responses. If you use the measurement of and say, give me gamma interferon responses, you get all gamma interferon responses, irrespective of how it was, whether it was ICS, ELISpot, or ELISA. So that can be very useful and very powerful if you're looking for correlates for protection, as an example. Then the allele finder. This allows you to look at the restriction, MHC restriction that was defined in the assay. We currently have alleles from 16 species, and that equates to many, many, many individual alleles. So you can look at the species level, you can look at the class, or the serotype level, or the allele level. This reminds me to remind you that a lot of times when you're looking for data, as Randy said, look at the broader picture, because sometimes you'll look specifically for an allele and you'll say it's not there, but that's because perhaps it's there under the serotype, not the 4-digit allele. So keep that in mind, because we capture what's in the published literature, not what we want to be there. So always keep in mind that sometimes it's up at the higher node, for lack of a better way of putting it. And so this allows you to peruse the menu of the different species we have data for. You can also use the tree to look by serotype, or by the molecule, or by the species. You can look at class. So that's pretty straightforward. All right, the host finder, I'm not going to spend a tremendous amount of time on this because it's really straightforward. 9 times out of 10, you'll have what you need on the main page, and you won't need to open the finder. But again, this will be useful perhaps if you're looking at a very specific or unique kind of mouse strain. And the disease finder. So this is the last of the finders as you're going down. The disease finder allows you to look for a very specific disease within the 4 broad categories. So you would click open the finder, and again, you maybe be able to type in asthma, and you'll be able to see the different types of asthma that we have captured thus far. You can also use the tree to look and see what's there, spend a little time. You can look at animal models. We have for under autoimmune disease, we have a branch for the animal models of autoimmune disease, so you get an idea there. So I think at this point, I'm going to be turning it over to Nima. He's going to be walking you through the specialized searches.
Good morning, everybody. Yes, my name is Nima Salimi. I'm actually also a member of the curation team. So this morning, I have the opportunity to continue to walk you through familiarizing you with some of our search interfaces. And at the conclusion of my slides, we will actually have a break. So, and on the other side of the break, the goal is to take it to the next level and get our hands on the database and do more live querying and actually utilize some of the feedback that you all have provided to us and touch on some of the topics that you've given, your areas of interest that you've provided. So the specialized searches are accessible here from the home search from a distinct menu at the top toolbar. And as you can see, there are many items there. There are actually, the specialized searches can be subdivided into 3 subcategories, if you will, of types of searches that one can perform. And these include detailed searches. So as you've heard, we capture an immense amount of detail where available from these manuscripts. And these detailed searches allow one to really query down to the field level. So every single database field that is in the database, one can set as a parameter to query. Excuse me, so literally every single field is available. And therefore, one, as I'll demonstrate, you can create very pointed queries, ask very specific questions. Secondly, there's an identifier search, which allows one to access data directly using some of the identifiers that you heard about this morning. including both identifiers that are inherent to the IEDB, but also these external databases with which we link can be queried directly there. And then finally, you've heard a little bit also this morning about 3D structures and cases where the data have PDB relations or PDB structures in them. And I will demonstrate to you the most direct way to access that structural data through our Browse by 3D Structure feature. Excuse me. All right, so I think one question to tackle off the top is, when might I decide that a detailed search is more applicable for me versus a homepage search? Well, as you heard this morning, we've taken great efforts to making that homepage search, tune it up for the best use of our user data community. And as Bjorn said, it's a work in progress, so we've refined it and continue to do so, and would love your feedback. But what we found is that the homepage search is adequate for 95%, more than 95% of queries that our users are performing. And in fact, it is being used for more than 95%. So we find it to be a very robust search interface, and as Carrie had also mentioned, you will probably find yourself, at least as a good entry into the database, using the homepage search. So you can see, for example, and this is not an exhaustive list, but there are a lot of things that you can do that the homepage search informs. However, there are cases where you might want to probe further details that the fields for those sorts of details are not provided on the homepage search. So for example, you've heard this morning about our jargon in terms of immunogen, antigen. If you wanted to ask in the context of a specific pathogen, for example, what's known epitope-wise in the cases when the host was only immunized with an organism or only immunized with that epitope, you're asking the question about an immunogen, and so that might be a very good place to probe the data using the detailed search. And again, we will make efforts to show you these, not only with these screenshots, but in live examples as we go. So going further into the detail search, when one chooses, beginning here with the epitope detail search, what you see is essentially like a screen that looks very familiar to what you saw with the homepage search. So along the left-hand side are a couple of panels here of epitope field-related search fields and reference-related search fields. But along the middle is that familiar view that is really unchanged from what you've seen earlier. So the tab organized layout of the different data. So at this point in time, if you choose epitope details, you're going to be given every single epitope in the database, and it's essentially your opportunity to delimit or filter or refine using these fields. So as you've seen this morning on the epitope detail search, you've got on the home page ways to define, for example, structural parameters or source parameters for your epitope, and as well as for your reference. And this is no different, except for you've noticed that the data headings have plus signs next to them, which implies that there are more fields buried within those. And in fact, if you get into the expansion of those fields, you see some of the breadth and depth of the types of fields that we capture. So things that, such as like the start and stop positions, when those are given in the data by the authors, we will capture those. You can see other instances of, so for example, we will capture comments. So very often we will clarify the data with comments that the curators put in. So literally down to the field level, these are available for you to query. We also have what's called an epitope-related object cluster of fields. And this basically has three choices from the menu. One's called epitope analog, epitope limitope is the second, and epitope neoepitope is the third. And what the related structure allows you to define is, we often come across data where the epitope is, for example, a non-naturally occurring sequence, peptide, for example. This might be because the authors of the study were doing amino acid substitutions to define key residues. But in those cases, the epitope itself will not be linked necessarily to a naturally occurring source. And so when one queries, let's say by influenza, if you have an analog of influenza, you may not retrieve that data because it's, again, not naturally occurring. So in just one example of how this data might appear and how you might get at it if you're interested, this is an actual record from the database in which this linear sequence has alanine substitutions in it, so it's an analog of this naturally occurring flu peptide, flu epitope. And so when we curate this data, we capture it true to the alanine substituted sequence that's given, and of course we cannot tie that to any naturally occurring sequence or any GenBank UniProt record. So we indicate that it's an analog of this naturally occurring hemagglutinin peptide, and we put the non-alanine substituted version of it there, so that if one wishes to capture this data, one way to accomplish that would be to actually query for analogs of flu. Excuse me. All right, so briefly, in the lower cluster of fields, which are the reference fields, you've seen this morning, Carrie demonstrated it, but again, we have the ability here for you to search by authors. Author last names is a very common search that users perform. You can delimit your search by journals. You can delimit the start and end date, for example, of publication. And as you've heard, we've got data that comes both from published journal articles and submissions. So if your query interest might be one or the other, you can set those parameters in your query and filter out, for example, everything but submitted data. So if you choose one of the other three detail searches, either TB or MHC, in addition to those two panels that I just briefly overviewed, you're going to see two additional panels, one called the host and one called assay. So again, intuitively, those are fields that collect or house information or details about the host and about the assay, but go beyond what we've seen so far this morning. So for example, as Randy had mentioned, we capture how the host encounters an immunogen, whether it be a natural exposure, deliberate administration in the case of an animal. So these fields allow you to define how the host was immunized, if you will. Furthermore, you can actually specify what that immunogen is. So the relationships that Carrie overviewed, for example, we might say that the immunogen was the source organism. What we mean by that is the source organism of the epitope. So you might be interested in seeing cases where mice, for example, were only administered with the source organism of the epitope. And here, as opposed to on the home page, you could define those parameters. You could also specify host diseases. On the assay side, this goes beyond just the readout, if you will, or the actual experimental assay. These are parameters in which the assay was performed or with which the assay was performed. For example, in the case of a T-cell detail search, you might specify, I'm interested in CD8 positive cells exclusively. And because when those are defined in the literature and the submission, we capture that, you could query by that and exclude, for example, CD4 responses. Similarly, on the B-cell side, you could specify, for example, I'm interested in solely monoclonal responses of the antibody versus polyclonal. You can query for specific isotypes as well and delimit your search that way. As we saw on the immunogen, sort of the mirror field or the sister field of immunogen on the assay side is the antigen, because this is like the chemical entity, the biological entity that was used to elicit the response either in vivo or in vitro, or both in some cases, and those antigens can be also specified. So I want cases where the host, the human, was naturally infected, but that the readout in the assay was done against an epitope only. So that would be a question that you could easily formulate and query by using the specialized searches. So to expand upon that a little bit, here what I've done is I've peered into the host fields to unveil these two fields called the in vivo process fields. So these go with the go back to the exposure or the initial contact of the immunogen. So when the host has encountered immunogen in vivo, that can happen once or it can happen twice, and we call those first and second in vivo processes respectively. So again, it might be deliberate inoculation of an animal in an animal model. The in vivo process may have been that the host was simply exposed or sick in nature. So in any case, you can see these have plus signs so that by expanding them, you're given the choices. And these choices are a set of checkable boxes that are predefined terminology that explain kind of what those processes are. So for example, and these are multi-select, so you can specify more than one if you so desire. But in the case where, supposing a mouse was administered with something, we would capture those under the administration group. So there are a few different administrations. A case where a living mouse was immunized, of course, would be an administration in vivo. Cases where a host, for example, a human host may have been diseased or have an occurrence of an allergy or an autoimmune disease, that would fit under the occurrence group. And then we have various exposures where there may have been, in nature, some sort of an exposure that is documented. That host may or may not be diseased, but in any case, you could delimit the return the results by hosts that are exposed. Some others that we have, as Bjorn mentioned early on in going along with the data that we capture, include transplant, transfusion. There are cases where there's simply no immunization and all the sort of pre-assay stimulation was done in vitro. And cases, very few, but some cases where we simply don't know or it's unspecified what, if any, exposures there were. So all of these process types are defined in our curation manual, but of course we're here to help you get acquainted with them as well and utilize them as you desire. So kind of a partner field with that you might find useful with the process type, as I've mentioned, is actually specifying what the immunogen might be. So I'm not only am I interested in how the host acquired immune reactivity, be it a deliberate inoculation or a natural infection, but I also want to specify what that inoculant might be or how what the exposure was to. So these are where we would use the immunogen and define by this drop-down list series of relations. So if, for example, you're querying for epitopes from dengue and you only want cases where the host was exposed in some way or injected in some way with virus, then you would choose, for example, epitope relation source organism. So as I previewed in the previous slide, the sister fields on the antigen side can also be delimited to be defined, and these equivalent drop-downs exist on the antigen, so you can say I want the antigen to be only epitope, for example. Okay, so moving ahead from the 4 detailed searches, I mentioned the identifier search at the top. So if you select that, you'll get this pop-up window that has this cluster of 7 identifiers. We've already used this in real time today when we did that KEBI ID search you saw earlier. So these are distinguished in 2 groups, the identifiers that are unique to the IDB and created by us. So these are internal identifiers that are generated as the data enter the database. So every epitope is given an ID, every submission and manuscript that we curate has a reference ID, submissions have IDs, etc. And then, as you've heard as well, we make extensive links and use of outside database resources. And so if there are data related to one of these, you can also query to that and gain entry quickly that way. So a brief example of that might be one real popular one is I want to find a paper that I've published and I want to see what you captured about it. So simply putting your PubMed ID there and searching will take you to the familiar summary screen where you have the data from that reference summarized in tabs, and then, of course, the left fields are the data query panels that you've seen earlier. So finally, from the specialized search menu, you have a browse by. So the browse by 3D structure simply implies that the data that have been taken from PDB structures. So whenever a PDB structure is reported in a manuscript, regardless of the scope of that data or what kind of subcategory it falls into, we make great efforts to capture those data and categorize them here. So they're essentially organized by the organism that's either the source of the antibody, the source of the TCR, or the source of the MHC. And so those are essentially made into a tree, a data tree that you've seen in earlier examples that are collapsed but can be probed and basically expanded. And all along the way, the numbers in parentheses indicate the number of assays that go with each of those branches. And of course, they update as you go down further. So here what I've done, for example, is I've gone under the T cell structure and drilled down to the HLA-A*02:01 allele. And you can see here we have 62 assays. So if this is clickable, so by clicking it, it will again come back to the summary screen. And these 62 assays are reflected in the assays tab. And so it was mentioned earlier that anytime there's a 3D structure, you'll see this icon on the assays tab. And you saw earlier that by clicking those, you can, trigger the 3D viewer. You're going to see a lot more of this over the course of the next 2 days. But the 3D viewer is essentially a JSMOL viewer where the user can manipulate this in 360 degrees in space and manipulate the structure and move it around. You can see color-coded down here, the chains are defined, both of the MHC and of the TCR. And in fact, the individual residues that may interact with the MHC, the TCR, both are specified so that the user has the ability to click and unclick. And just as an example here, what I've done is I've clicked all of the epitope to MHC residues. So here you can see that lighting up in blue, the lighter colored structure here is the epitope, but overlaid on top of those are the actual residues of the epitope. So you can individually lift residues on and off, superimpose them, and manipulate this in space. So this is very customizable, and this image is also exportable. So that's our 3D viewer, and with that, I think we have time. We have some time before break, right?
We do have some time for questions.
Questions.
Yeah.
Be happy to take questions. Carrie and I can take your questions because we don't have break scheduled for a while. Please.
Your HLA molecules, are they standardized to 2 levels of resolution on your database, or can they be 1 level of resolution, or how is it done?
So the question was, are HLA molecules, are they standardized to any specific level of resolution? So we take what the paper states and literally capture it as such. So if a paper says this was bound to an HLA-A2 molecule, we literally have a selection that says HLA-A2.
Does it say A2 or 02?
Either. I mean,
Whatever the author says.
whatever the author says.
Whatever the author says. So
Yeah.
the early papers would have A2.
Correct.
You don't translate that to O2. The
We
more
don't
modern
translate it in the input side.
You're literal from the paper.
We're literal from the paper. So now, in the trees, if you did an O201 structured query, you would retrieve a data point that had been listed as A2.
Oh.
Because we do that logical connection on, the database side.
So
Bjorn, go ahead.
No,
There's 3D structures.
we don't have an A02 entry.
No, but we have A3.
We have A2.
We have A2.
A2 is a serotype. That's based on, so serotyping is based on antibody binding patterns. That's a different level of resolution. We don't have A02, which doesn't exist at all. It's like a really weird concept. So A0201 is a completely defined protein sequence. A2 is an antibody binding pattern. A02 is a grouping term that may or may not somehow correspond to a certain set of which we're not using.
The 3D structure is 4-digit.
Every time it's if you know the protein, then you can always do a 4-digit resolution.
For image to binding assay on cells or something. Or even ligand elution, sometimes they say we eluted from class 1. They don't tell you anything else.
Yeah.
No, but he asked specifically about A02. He was asking about these grouping things which are shouldn't be used by anyone.
So, but the A2 will pick up the 0201 as well in your database?
Yes.
That's where
Yes.
And the 0201s, will they pick up the A2s?
No.
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 So it won't go that direction?
No, specific to general.
General specific.
Yeah, one way we will do it, we won't do it the other way, right?
It's a treat.
So
It's a treat.
we always have the database was designed to capture what we encounter in the literature, which is vastly variable in some instances. Sometimes it's very consistent. And then there's what people typically report. So we're always vacillating. So when we get questions, sometimes we confuse the answer we're giving you. So how things are categorized. We'll try better to ask more specifically what you're getting at, so that we don't befuddle you with our answers.
Yes, please.
I have a question, I think, for Carrie.
Yeah.
When you were going through the TCR query page, you showed like
The receptor?
you
Yeah,
showed just
yeah.
like a listing of different alpha betas. Like, what do you do if you have both of your VDJ loci rearranging, like on your alpha or something like that? So you have a T cell receptor that has an alpha and a beta pairing, and then an additional alpha that's paired to the same beta. Are you able to account for that? And then, I guess, are you able to take in sequences from like papers that are just giving the beta, or is it like you're only incorporating into the database paired alpha and paired beta?
We capture, again, we're back to good questions. We capture what they report. Sometimes they report a single sequence, sometimes they report full characterization. So we capture what they provide. But the receptor, the interface should allow you, yes, so here we go. So this should allow you to get at the chain or the region that you're interested in. If there's been more complex scenarios that would probably analyzed at the level of, once you've downloaded the data, you can look at the data and analyze it that way. The complexity of what is reported, how do I put this? What the scenario that you define can't be searched for using this interface, but it can definitely be analyzed once you download the data and you can look at the data, look at the sequences and stuff like that. And Randy does a lot of this and has set up all of the data tables for this, so she's an expert in this.
So if they report paired TCRs or antibody chains, we keep them paired in the database. If they report one of the, alpha chain or beta chain or heavy chain or light chain, we enter whatever they give us.
Okay.
And then you can search for CDR3s.
Right.
And you can specify if you're only interested in beta chain, you can do that. But if it was paired, you'll know when you see the
Okay.
results.
I have a comment on that. So actually, if like most of sometimes you of course authors provide only beta or alpha CDR3 sequences. That's correct. And if you want to get more information just based on the beta CDR3 sequence, for example, we have actually grouped the receptors based on CDR3 sequences. So if you're interested, you can either talk with me, Randy, or Bjorn about this. I'll be happy to give you.
That's Swap Neil, he's one of our informaticians. Okay.
Yes, I have a curiosity regarding receptor data. And you showed a slide and I could see here PCR heavy. VCR heavy.
Swap now.
I didn't hear your
No,
question,
no,
sorry.
in the slide that you show.
Heavy chain.
Yeah, heavy,
Oh,
heavy.
yeah.
VCR heavy. I mean, I get VCR heavy light, VCR heavy, and then you have VCR light. What type of composition is that?
Sometimes the Campbell antibodies are reported as heavy, although they might just provide just one chain because it's a homodimer. So we have reported them as heavy, and light can be constructs, basically engineered constructs from the authors. So we report them based on the chain types. That's why we have a light and heavy categories.
So can you have a specialized search where you have the structure, the 3D structure, and the binding affinity? Like the outputs at the same time?
You can do that from the
You can.
yeah, the main page, yeah. And we'll be showing you, I'll be showing you that in the afternoon, so,
Oh.
yeah.
For non-peptidic epitope finder, can smart structure be searchable?
I'm sorry?
Can smart structures be searchable?
Smarts.
If you download the data, the Smiles is there,
Smarts.
but.
Oh, Smiles, I'm
But
sorry.
not in the
Yeah.
search interface.
Yeah, and I can show you, I'll show you on a details page. Which do you have, center page?
But you can also go into Kebi and put your Smiles in
Yeah.
Kebi, get the Kebi, and then search the Kebi.
Sort of like, right. But we actually are aware it's very hard to search non-peptidic molecules. That's something we have on our to-do list to try and find alternate ways of searching. Because if you have a name like penicillin or nickel, it's pretty easy. If you have carbohydrate, it's hard to find the data.
How to differentiate response due to natural infection versus cross-reactive? Cross-reactivity due to some other similar, like if you take dengue virus infection,
Yeah.
so there is some, like in infected people, like they give some response, but there are co-circulating other flaviviruses also.
Mm-hmm.
So whether it comes from cross-reactivity due to those.
To do the search, you can search it on the high node for flavivirus, for example, is that what you mean? Yeah, so you can look at T-cell responses in flavivirus, and you can look at, and I'll show you, in fact, I'm gonna do a demonstration of dengue after the break, before lunch. And he's asking how you can look at different, besides dengue, other viruses within that group. And I can show you how you can look at that, and you can look at, because it'll display it on the antigen tab by protein from the virus that it comes from. So I can show you that. Yeah. Is it time?
Yeah.
Yeah,
So
all
real
right.
quick, I'm gonna cut everybody off here. I did forget to do one thing in the morning that might help as well. And so really quickly, I'm just gonna have the LJI IEDB team stand up, and so we can see who they are. So you don't just have to find a name tag, you can see a face, right? So we've already had Nima and Carrie present, so Nima and Carrie. We have Randy right here, who also already presented. We have Leora, Ellie, Sinu, Debbie, I'm gonna make you stand too. We've got Debbie, Bjorn in the background. We have Sinu, I skipped Sinu. Sinu's right there, Jason, and then we have Sandeep and Swapnil in the back corners. So you know who we all actually are now, so you don't just have to look for the color differentiation on our name tags. We're gonna take a quick